Hello, IB Bio students. So today we're going to continue with the process of glycolysis and fermentation. So let's start with the essential understanding here that says that in glycolysis, glucose is converted into pyruvate in the cytoplasm. So the main organic compound that we use in cellular respiration, we know is carbohydrates. And although lipids and proteins can be used, they are not ideal. The reason for this is because lipids, even though they could provide more energy per gram, they are harder to transport and digest. And proteins are not used as the main energy source because they could potentially release toxic nitrogenous compounds when broken down. So for that reason, the main organic compound that we use to yield energy is carbohydrates, and glucose is that main molecule. So we have to think of glycolysis as the first step in cellular respiration, and we know that cellular respiration is going to yield a controlled breakdown of this carbohydrate known as glucose, and it's going to generate energy. So where does glycolysis happen? It happens in the cytosol, which is the name for the cytoplasm of the cell. And this is the first step that happens in glycolysis. We have a 6-carbon sugar, which is a hexose sugar, and we break it down into 2 molecules of pyruvate. So notice that we're taking a 6-carbon sugar and we're breaking it down into a smaller 3-carbon sugar, and it's 2 of those smaller 3-carbon sugars. Now there's many intermediate steps, but we can summarize glycolysis into 4 key events. So let me move this down, and here are the 4 key events of glycolysis. So glycolysis has a process known as phosphorylation, lysis, oxidation, and ATP formation. So what happens in phosphorylation? So in phosphorylation, you're going to take a hexose sugar, which is glucose, and you're going to phosphorylate it. And remember that when you phosphorylate something, you add a phosphate group. So when you phosphorylate the hexose sugar by 2 molecules of ATP, you're going to form a hexose biphosphate. So let's look at it here in this slide. Here we see the glucose molecule. If you add 2 ATP molecules to provide the phosphate groups, right here is the process of phosphorylation. So what you're going to end up with is now a fructose 1,6 biphosphate. So this glucose becomes phosphorylated, and this is what you get. So fructose is an isomer of glucose, and notice that they are still 6-carbon sugars. So this is the first intermediate in the phosphorylation process. So going back, what happens after phosphorylation? Lysis happens. The hexose biphosphate, or the fructose biphosphate, which is still a 6-carbon sugar, is split into 2 triose phosphates, or 2 compounds of 3-carbon sugars. So here we see it here. We see the fructose 1,6 biphosphate, it's split. Why? Because remember that the phosphorylation process makes it unstable. So this molecule can now split into triose phosphate, and triose phosphate is a 3-carbon compound. So what happens after lysis? Oxidation happens. That means that hydrogen atoms are removed from each of the 3-carbon sugars, and this is what we called oxidation, to reduce NAD plus to NADH. So that means that 2 molecules of NADH are produced in total. So we're going to see oxidation right here. So here we see that NAD+, which is an electron or hydrogen carrier, is going to pick up that hydrogen here found in the 2 triose phosphates, and NAD+ is going to get reduced into NADH. So that means that in that process, you're going to release pyruvate plus a phosphate. And here you see pyruvate plus a phosphate. So that means that in this process here, you're going to release 4 ATP molecules when you get the formation of the 2 pyruvates. So this is the 4th step, right? ATP formation. Some of the energy released from the sugar intermediates is used directly to synthesize ATP. the direct synthesis of ATP is called substrate level phosphorylation because you need a sugar intermediate. And you're going to end up with a total of 4 molecules of ATP generated during glycolysis. So this is important, 4 molecules of ATP generated during glycolysis by substrate level phosphorylation. But this is not net molecules of ATP because remember that it took 2 molecules of ATP to start the glycolysis process. So then that means, and this is the key takeaway here, that you get a net total of 2 ATP molecules in glycolysis via substrate level phosphorylation. Okay, so what are you going to get at the end of glycolysis? You're going to get your 2 molecules of pyruvate, which is a simple carbon compound. You're going to end up with 2 hydrogen carriers that have been reduced via oxidation, and you're going to end up with a net total of 2 ATP molecules that have been produced, right? Because it took, even though you produced 4, it took 2 to drive the cycle, so that means you get a net total of 2 ATP molecules. So if we look further down here, we see the overview of glycolysis. Here we see the 6-carbon compound, which is glucose. You require energy, right? 2 ATP molecules to get the breakdown of the 3-carbon triose phosphates. These are intermediates here. Phosphorylation happened here as well. And you're going to release 2 hydrogens here via the oxidation process. You are going to reduce NAD plus into NADH plus H plus. You are also going to release 2 ATPs and 2 ATPs here via ATP production. And you're going to end up with 1, 2 molecules of pyruvate. Now the net gain again is 2 ATPs, 2 NADHs, and 2 pyruvates. So that's where you're going to end up as a net result of glycolysis. So moving further down, what happens next? So now that we end up with a small net gain of ATP without the use of oxygen, what's going to happen next? So this glycolysis, remember it happens in the cytosol and it does not require oxygen. But depending on the availability of oxygen thereafter, pyruvate may be subject to one of the 2 alternative processes. So pyruvate has a choice now, right? If aerobic respiration occurs, that means that there is oxygen and you can continue with cellular respiration eventually into the Krebs cycle and the electron transfer chain to produce more ATP molecules, a total of 34 more, plus the 2 in glycolysis giving you your 36 ATPs. But if no oxygen is present, then anaerobic respiration happens, which we can also refer to that as fermentation. And this occurs in the absence of oxygen and no further ATP is produced. So the other videos are going to focus on what happens if aerobic respiration does occur, right? The link reaction is going to happen, the Krebs cycle is going to happen, and the electron transfer chain is going to happen, and you're going to get a whole bunch more of the ATP molecules. But if anaerobic respiration happens, then we see that there's no oxygen present, pyruvate is not broken down further, and no more ATP is produced. So we call that incomplete oxidation. So what happens to that pyruvate that remains in the cytosol? Well, it's got 2 pathways, right? If you're an animal, that pyruvate without oxygen is going to be converted into lactic acid. And if you're a plant or a yeast, that pyruvate in the cytosol is going to be converted either to ethanol and to ethanol and carbon dioxide. Now, it says here that the conversion is reversible and it's necessary to ensure that glycolysis can continue to produce small quantities of ATP. So if we look at the fermentation process even further, it tells us the pathway, right? Here we see the starting with glycolysis, the 6-carbon sugar. Here we see a phosphorylation process. Here we see that after lysis and ATP production, we end up with 2 molecules of pyruvate. Now, if there is no oxygen present, right, if there is no oxygen present here, we go into anaerobic respiration, otherwise known as fermentation. And what happens to these pyruvate molecules? Well, if you're a yeast, you can or some sort of plant, you can produce ethanol, which is a form of alcohol. You're also going to release carbon dioxide, and this is why bread rises, right? If you're baking bread. And if you're an animal cell, a muscle cell, and you run out of oxygen, then you are going to go into anaerobic respiration or fermentation, and you're going to produce lactate or lactic acid, also carbon dioxide. And remember that lactic acid is the byproduct of when your muscle cells run out of oxygen. And that's why you feel your muscles aching the next day after a rigorous workout. So that was glycolysis and fermentation. So make sure you review both your BioNinja notes. The slides here and in thinking are also very good at showing you the visual of what happens during glycolysis. And that's it for today.